Breathing is essential to life. As with every breath, the body is replenished and cleansed, a process made possible by the lungs. Every year, in the US and Canada alone, almost 2,000 babies are born with lungs that are too small for them to breathe properly. Tragically, half of these babies die and the other half have severe difficulty breathing. These babies suffer from a condition called congenital diaphragmatic hernia, or CDH, as they are born with a hole in the diaphragm, the muscle that separates the chest from the belly. So the organs that are supposed to live in the belly, such as the intestines, slip up through the hole into the chest and squash the lungs to a point that they don't have enough room to grow. Even when we do surgery to close the hole in the diaphragm and return the bowels into the belly, the lungs remain way too small. We all agree that a therapy for these tiny lungs is desperately needed. Once the baby is born, it's too late to make the lungs grow. The good thing is that nowadays, we can recognize these babies with CDH before birth with a simple ultrasound scan. So we all agree that the ideal timing to intervene on these lungs is when the baby is still in the mom's womb. However, many research groups have tried, but none of the treatments tested so far has been successful. I'm very excited to tell you that we think we have found a solution in the amniotic fluid. For years, we've been studying stem cells from the amniotic fluid, and we have now demonstrated that they release small droplets called exosomes that can make these tiny lungs grow bigger. Exosomes are like postmen who carry messages from a cell to another. They're released by all cells, but those coming from the amniotic fluid are special, as they can regenerate organs like the lungs of a baby. In fact, they carry a message made of genetic material, the so-called microRNAs, molecules that tell the recipient lung cells to restart growing. When in our studies we gave exosomes to tiny lungs, we observed that they restarted growing and functioning like normal.
When we colored the exosomes to see where they were going, we found them inside the lung cells and saw that they released their cargo. This caused the lungs to make more and more of the molecules that help us breathe, like surfactant. And we saw that the treated lungs were more mature as they had more specialized cells.
I'm very excited because this is the first fetal therapy using exosomes. Our study opens avenues to test the use of these exosomes for other congenital anomalies. This means increasing the chances for millions of babies to survive and live a normal life.